When getting data into TameNMR to visualize NMR spectra, it's important to make sure that the format is correct for the data to be viewed properly. To pull data into TameNMR, select Get Data from the left-hand browser window and Upload File. Here you'll be prompted to drag files over. The files we're going to import are Bruker format data files, as you can see here. These were downloaded from the MTBLS 541 dataset hosted on EMBL EBI metabolites, and in here we have 72 spectra. These spectra have then been selected and compressed in zip format into two folders, cell.zip, which has 28 spectra, and cell_tumor.zip, which has the full 72 spectra. It's important to note that the title file within the format of Bruker must be individual for each sample, otherwise the import will not work. So if we take our zipped folder and drag it over to the drop files here, we can click Start to begin the import. This process might take some time depending on how many files you're uploading. Once uploaded, you will see they appear in your history window, which is the right-hand browser, going yellow with a spinning disk whilst uploading and green on a successful attempt. If the file for some reason goes red, that means there's been an error in the upload and you want to check our troubleshooting guide to make sure you haven't made a mistake. Once the data has been uploaded, it must then be imported using the import data command. Here we select the data source as data and the zip file we wish to import before clicking execute. Again, the right-hand browser will show that the data is waiting for upload and import, going yellow when the process is active and green when the process is successful. Once you have your imported NMR data, it can be visualized by clicking on the eyeball, and you can see that your data is aligned with samples in columns throughout the spectra and the title that came from the Bruker file used as the header. TameNMR project has been funded by a BBSRC grant to the University of Liverpool. This is a collaboration between the NMR Metabolomics facility and the Computational Biology facility here at the university. For more information, please contact us via Twitter or access other tutorials in this series.